Welcome to Quantum Frontiers. Today we're tackling a cutting-edge topic at the intersection of physics and biology. Can effects like coherence, tunneling, and entanglement explain biological processes? Quantum coherence in photosynthesis enables energy transfer at nearly 100% efficiency, even across disordered molecular networks. Doesn't this demonstrate that quantum mechanics is central to biological processes?
Coherence plays a role, but it's highly context dependent. It works in tightly controlled protein environments and relies on vibrational coupling. At room temperature, the coherence lifetime is in femtoseconds. Replicating such delicate conditions in practical systems remains an enormous challenge.
Advances in biomimetic systems show promise. Synthetic frameworks are designed to mimic vibrational modes that stabilize coherence. Isn't that a step toward replicating nature's quantum optimization?
It's a step, but biomimetic systems operate in simplified conditions. Real biological systems are noisy, chaotic, and governed by constant thermal fluctuations. Have these systems been tested in vivo, where noise would destroy coherence almost instantly?
That's where quantum-enhanced microscopy becomes crucial. Techniques like quantum correlation imaging allow us to observe nanoscale quantum dynamics in real time, even in living cells. Isn't this a breakthrough in bridging theory and experimental biology?
Observing is important, but it's only one piece of the puzzle. Detecting quantum effects doesn't mean we can use them. Stabilizing coherence or entanglement in noisy environments is a massive, unresolved issue. Have we even achieved sustained coherence in a single biological reaction?
Sustained coherence is a challenge, but materials like topological insulators offer a pathway. They're inherently resistant to decoherence, even in noisy environments. Couldn't they help stabilize quantum processes in biological systems?
Theoretically, yes, but biological environments aren't just noisy. They're chemically reactive and thermodynamically unstable. Topological insulators might work in a clean lab, but in a biological setting with fluctuating ionic concentrations and chemical reactivity, that's uncharted territory.
Let's pivot to tunneling in enzymes. Experimental evidence shows that quantum tunneling is critical in certain reactions, like hydrogen transfer in alcohol dehydrogenase. Isn't this a clear case of quantum mechanics driving biological efficiency?
Tunneling is intriguing, but it's not universal. Most enzymes function perfectly well under classical models of catalysis. You're highlighting a specific case and extrapolating it to all enzymatic processes. Where's the broad evidence?
Classical physics can't explain enzyme activity under extreme conditions or the speed of proton-coupled electron transfer. Isn't tunneling the most consistent explanation for these anomalies?
It might explain specific outliers, but that doesn't make it the dominant mechanism. Protein dynamics, substrate binding, and induced fit models offer equally plausible explanations. Have quantum proponents ruled these out rigorously?
Quantum-enhanced Raman microscopy is already uncovering molecular behaviors that classical tools couldn't detect. Isn't this evidence that quantum tools are transforming how we study biological processes?
Transforming detection, yes, but detecting tunneling or coherence doesn't mean we can engineer it for biotechnological purposes. The gap between observation and application is immense.
Isn't this the same incremental process that led to breakthroughs in quantum computing? Progress comes step by step, building on early discoveries.
Quantum computing succeeded by solving fundamental challenges like decoherence and scalability. Has quantum biology addressed those issues? Where's the roadmap to practical applications?
Fair point, but bold experimentation often drives breakthroughs. Without taking risks, we risk stagnation.
Risk is necessary, but unchecked hype can undermine the field. If we oversell preliminary findings, we risk losing credibility and funding.
Speaking of preliminary findings, what about enzymes showing enhanced diffusion during catalysis? The diffusion coefficient changes depending on whether the substrate is present. Theoretically, enzymes are too small to alter their diffusion significantly, even in the presence of a substrate. How do you explain that?
That's one of the most puzzling phenomena. Classical physics would predict no change in diffusion coefficient due to the enzyme's size. Reaction-driven forces might explain it, but that doesn't address the underlying mechanism.
Some suggest that quantum forces, perhaps arising from reaction-driven tunneling or coherence, could contribute to these effects. Could enzymes be using quantum effects to drive their own motion?
It's possible, but extremely speculative. Enhanced diffusion could arise from thermal effects, conformational changes, or even hydrodynamic interactions. Have those been ruled out?
They've been explored but not conclusively ruled out. What about chemotaxis-like behavior in enzymes? They seem to move up substrate gradients, which defies classical explanations given their size and lack of active motility.
Chemotaxis-like behavior is fascinating but poorly understood. While quantum effects might play a role, simpler explanations like reaction, diffusion, coupling could also fit the data. Without direct evidence, it's premature to attribute this to quantum mechanics.
Isn't the presence of these anomalies a reason to investigate quantum principles further? Biology clearly operates beyond what classical physics can fully explain.
Investigate, yes, but jumping to quantum explanations without ruling out classical alternatives risks overhyping the field. We need more data before making grand claims.
That's all we have time for today. Is this the next great scientific revolution, or are we still far from realizing its potential? Thank you to our guest for the rigorous discussion, and thank you to our listeners for tuning in. Until next time on Science Frontiers, stay curious, stay critical, and keep questioning.